Hello and welcome to the next episode of the Psychedelic Medicine Podcast. I'm Dr. Lynn Marie Morsky, your guide on this journey, and this week we're going to be discussing understanding psychedelics and neuroplasticity with Dr. Robin Carhart-Harris. Robin Carhart-Harris is the Ralph Metzner Distinguished Professor in Neurology and Psychiatry at the University of California, San Francisco. Robin founded the Center for Psychedelic Research at Imperial College London in April 2019, was ranked among the top 31 medical scientists in 2020, and in 2021 was named in Time Magazine's 100 Next, a list of 100 rising stars shaping the future. Now, before we get to Dr. Carhart-Harris, just a reminder that the Psychedelic Medicine Podcast is for educational and informational purposes only. Nothing here is to be construed as medical or legal advice. And one last reminder, as always, if you are a clinician and your patients are coming to you asking questions about psychedelics and you would like to educate yourself to be more prepared to answer those questions, please check out the Psychedelic Medicine Association. It is our mission to get you educated and equipped with the information you need to have those conversations with patients and make referrals when appropriate. And you can find us at psychedelicmedicineassociation.org. Now, without further ado, thanks so much for joining us today, Robin.
My pleasure. Great to be with you, Lynn Marie.
I'm really looking forward to this chat. Neuroplasticity is one of my favorite topics in the psychedelic realm. And so I'm extra excited to have this chat with you. And I'd love if you could just start us off by telling the listeners what neuroplasticity is.
Sure, happy to. It's a funny old thing when we talk about neuroplasticity. We're talking about the brain and its ability to change, essentially. So neuro, we're alluding to the brain, and plasticity, people should know in its most basic sense, means changeability. So in a sense, it's not necessarily actual change. That's kind of how we measure it, some kind of change, but it's a slightly different, but importantly different thing. It's changeability, the ability of something to change. So not change per se, but the ability for change. And yeah, that's the most basic definition of plasticity. If you were to look it up in the dictionary, you would see that. It might say something like the ability to be shaped or molded. So changeability is what plasticity is. And then neuroplasticity, that's the ability of the brain to change.
Okay. And how is neuroplasticity related to mood disorders like depression and anxiety or substance use disorders, something like that?
Well, that's a great question. Of course, we don't have it entirely nailed down, but one of the most reliable findings in biological psychiatry is that stress atrophies the brain. So whether it's non-human animal research, looking at neurons themselves and how they atrophy, their different components of neurons, like the branchy bits, the so-called dendrites. When a neuron is healthy, a little like a healthy tree, it's got a lot of branches, a lot of leaves, a lot of abundance there, it's thriving. But under stress, that branchy abundance withers away. And so you see that in non-human animal research, like rodent research, and even more specifically, mouse research. You might look in a mouse after a period of chronic stress, that poor mouse undergoing some paradigm that's caused it, significant ongoing stress and presumably distress. You look in its brain at the health of its neurons, and they're not very healthy. They've atrophied away. So it's super reliable in terms of animal research. Then in humans, we can't operate with such invasive techniques that, we stress a human, kill it, God, look in its brain and see how healthy or otherwise it is. So instead we use brain imaging, and we look at things like, gray matter density, cortical thickness. And in humans, we don't intentionally induce stress, or within reason, we might do that in like acute stress studies. but we don't induce chronic stress. That would be unethical. So instead, we might look at people where we assume they may have undergone a period of adversity or stress. So maybe people with PTSD, or maybe people reporting high adverse childhood experiences. And then if you look in such people, you do see actually that gray matter's down in the cortex, it's decreased, cortical thickness is decreased. So, across species, stress, distress, trauma, adversity is bad for the brain. So we know that, and we also know that this, and what I mean by this, stress, trauma, adversity, probably more than anything else, more than genetic contributions, for example, is a major, maybe even the major contributor to mental illness. So you put two and two together, and it's kind of easy to surmise that stress is going to cause brain changes that are going to produce a particular as jargon, but sort of phenotypic presentation or trait level psychological presentation that could look like mental illness. Yeah. Yeah. And so if that's the picture, stress has been bad for the brain, pushed it into unhealthy ways of looking in terms of its anatomy, and we presume functioning, and there's some evidence to support that too. Then maybe it's been pushed in a direction of getting stuck in ruts. Maybe certain circuitry's been overly reinforced. So in a funny sense, that's plasticity too. We might call it even something called Hebbian plasticity. People might be familiar with the phrase cells that fire together wire together. And it's this principle that repeated associations tend to reinforce. So whether by sort of selective atrophy and the pruning away of options, connections, or the selective reinforcement of circuitry, say in drug addiction, repeated use of a highly rewarding drug, you're shaping the brain in a particular direction. I call it canalization. Well, it is called canalization. It's a phrase and a idea that goes back to Henri Bergson in 1911, but it means getting stuck in ruts. So that would be an example of, in a sense, unhealthy change, carving ruts in the mind and brain and behavior that could do with some kind of healthy change, some healthy neuroplasticity, maybe something regenerative to take you back from that carved, problematically carved picture of things. Yeah, so I know there was a heck of a lot there, but I think I took us to some key stuff in terms of why plasticity, and here's a bit more jargon, homeostatic plasticity, a kind of reset or recalibration, might move you towards health, or back away from illness and back towards health. Yeah.
Well, I'm very excited that you brought up the relationship between neuroplasticity and canalization, because I, at some point in time in this interview, wanted to ask you about the first thing that I knew about you before we met years ago was that you were the snow globe person, right? You had come up with, like, you had made this metaphor for what was going on with psychedelics and the default mode network. And I was so excited to like throw that at the end and ask you about it. But now I'm and in the papers that I read relating to this, and I'll have you give that metaphor in a minute if you will, but I thought it was only related to entropy and new connections. But now I'm understanding that this canalization, and is that the same, like the snow globe grooves? Is
Yeah,
that the same thing?
it
Okay.
is the same thing. So yeah, it's true. I'm the snow globe guy, and that was a magazine interview with, the magazine was Nature, and the journalist was asking me to theorize about psychedelics as a treatment for addiction. And I said it's like shaking a snow globe. That's how I imagine it. Um, the pathways get overly reinforced in addiction, like this thing, methamphetamine, crack cocaine, whatever, heroin, uh, is the thing that you want that'll bring you relief, that is rewarding in a sense. And that circuit that kind of governs and dictates that strong association is overly strong, it's overly reinforced. And so the solution to that problem would be a big shake-up to actually weaken connections rather than strengthen them, weaken the excessively strong connections. And so I use this metaphor of shaking a snow globe. I know it's kind of abstract, but I think people like it because they can imagine it. They can sort of see the chaos, see the entropy, and intuit that that's going to re-level the surface. That would be homeostatic plasticity, which is like a recalibration, like a return to factory default settings. That is, here we're playing with a lot of analogies and jargon, but here's another analogy, first popularized by Norman Doidge in his book, something like the brain that changes itself, I think, of a snowfall. I only sort of discovered this after the sharing the snow globe thing, but a snowfall will give you a fresh coating of snow. So some fresh powder. My former PhD student, Mendel Kalin, also shared this analogy. This fresh coating of snow is going to give you freer opportunity or greater freedom of movement if you're, whatever you are, a skier or a hiker, to move in that space. The opposite of a fresh coating of snow might be really sharp ravines in an ice scape where you could slip in and get stuck and struggle to get out. Sounds like depression or many other psychopathologies. Too easy to fall in, maybe treatment resistant depression, too easy to relapse and fall in, and so hard to get out of. So you can sort of intuit how this freedom of movement is, generally speaking, a healthy presentation. Yeah.
Yeah, that makes a lot of sense. And could you elucidate for the listeners the difference between neuroplasticity and neurogenesis, or maybe just describe the different ways that neuroplasticity can manifest?
Yeah, I mean, neurogenesis is a specific thing that means growing neurons. And we have so many neurons, I forget the stat, but a lot. And in our brains, in our cortex, especially, and also in our cerebellum, funnily enough. Um, but we have most when we're little babies. And yet, we might be closer to God as little babies, as William Wordsworth would remind us. But in an analytical sense, we ain't that smart, can't do math and so on, can't talk most of the time. Um, so well, there's a heck of a lot of abundance there and freedom, but not much sophistication, you might say, not much analytical sophistication. So, sheer abundance it isn't always going to confer great functionality. I'm trying to remind myself of the question now. Sorry.
Oh, neuroplasticity versus neurogenesis.
Yeah, so neurogenesis as a born human, you don't really do. Like all the abundance and all the neurons are there from the beginning. And from there on, it's mostly pruning. It's mostly actually canalization in a sense. It's like selective carving of patterns of activity and connections that are going to serve you in some way, that serve a bunch of different functions, a repertoire of different functions. And that's the picture that develops as we develop, in Wordsworthian terms, it's like the prison house that forms, is that selectivity that develops, that modularity that develops as we develop. So neurogenesis doesn't really happen very easily at all in a born or certainly an adult human brain. The jury's out on whether you can actually grow new neurons. Maybe, probably, certain circumstances, but it ain't an easy thing. It's much easier to develop new connections, and that's been demonstrated in a bunch of mostly rodent neuroscience studies, looking at mouse brains and looking at the branchy stuff, the dendrites of the neurons, and how, in a sense, more branches grow, or a bit more specifically and accurately, it's more like the leaves, the spines on these branches. Their density increases after exposure to psychedelics, and that's been shown with a bunch of different psychedelics. With ketamine, it's quite short-lived. With drugs that produce longer trips, generally speaking, they produce more, we call it spinogenesis or synaptogenesis, the growth of these not just branchy bits, but leafy bits that last for longer if the trip is longer. Generally speaking, that rule holds. There might be some interesting exceptions to it, like 5-methoxy-DMT is a short trip, but may produce quite a lasting synaptogenesis. So that's a difference between anatomical neuroplasticity, the growth of branchy leafy bits,
I love it.
versus neurogenesis, which is really hard to do, which is actually growing new neurons. Generally speaking, that doesn't happen in a born human brain. All of the neuron growth and sort of pre-neurons, like neural stem cells, they're being grown and proliferating in utero, yeah, before we're actually born. So, um, yeah.
Because I thought I'd seen some studies on DMT where they were, at least in vitro, had
Yes.
yeah, had looked into whether that was able to spawn some neurogenesis.
Yeah, I think you're right. I think it's in vitro. I think it's hippocampal cells in a dish, and specifically in the dentate gyrus, a subcomponent of the hippocampus. Maybe Jordi Rieber was involved in that. So it was a super cool finding, but it was in a dish.
Yeah.
Yeah, so this is where we are with a lot of things. There's an exciting idea that psychedelics could reduce neuroinflammation. That would be exciting, would imply that psychedelics could be helpful for things like traumatic brain injury and so on. Inflammation's up in a bunch of mental illnesses as well, like depression and so on. So it'd be a great thing if it was true. It's never been shown. And the reductions in inflammation that have been shown are typically either in the periphery, so outside of the brain, or in things like, looking at cells in dishes and so on. So yeah, we just have to manage our expectations and understanding of what actually has been shown so far. Yeah.
Yeah, absolutely. Well, do you know how it was first hypothesized that psychedelics may impact neuroplasticity?
That's a great question. Yeah, where would that go back to? Um, yeah, I mean, I think Brian Roth is kind of legend in the psychedelic research space, really an expert on receptor pharmacology, done a lot of pioneering research there. I think he did a study that had shown um, synaptogenesis with psychedelics, but that was a period just outside of the window of the current, you might say post Michael Pollan effect psychedelic renaissance. So the person who really caught that wave, you might say, would be David Olson at UC Davis. Has done fantastic work, and no person is an island. Many would be listening, thinking, "What about me?" And sure, but so there was work done after, or alongside the Michael Pollan phenomenon. In 2018, he published his bestseller that really popularized that phenomenon. And what made the difference? I suspected that this would happen actually, because I knew some classic synaptogenesis work with cocaine, showing that branchy stuff grows after cocaine use in in reward centers of the brain. It's really classic work, published in top journals. And so I got me thinking, someone could look at psychedelics and how they impact branchy stuff in the brain. And so, opportunists, smart people caught that wave and did that work. David Olson and company are good examples there. And the difference between what cocaine does, one of the differences, and what psychedelics do is that cocaine promoting synaptogenesis was in these reward centers. Um, and psychedelics, it's in the cortex. It's in the cortex. It's in the aspect of brain that humans have so much of relative to other species. And it seems to be especially the case, I think this is true to say, in high level cortex. That's hard to look at in a mouse because they don't have that much cortex. So we have to extrapolate from the highest bit of their cortex, the most sort of association related bit of their cortex that might be something like a homologue or a version of the human prefrontal cortex, for example, massively expanded in our species. Um, but that's where you tend to see the synaptogenesis in the non-human animal studies, is in the highest aspect of the brain, their version of the frontal cortex. Yeah.
Okay, thank you. Now, for listeners, we're going to get a little bit in the weeds here because we would be remiss to have Dr. Carhart-Harris on and not get a little bit into the details of what's actually going on. So, in this paper that you and your colleagues released earlier this year, you went into the mechanism of action and you had divided into MDMA, ketamine, and the classical psychedelics. Would you be able to give us just kind of a top level view of how each of those leads to or impacts neuroplasticity?
Well, I'll give it a shot. I mean, the main thing with ketamine is that the window of increased plasticity is brief. And that's kind of nice, or that makes sense, because it reflects how ketamine seems to work therapeutically, that it provides relief somewhat short term, unless it's twinned with, say, psychotherapy, or you do repeat administration and get someone out of the rut that they were in. And then, in a sense, it's sort of up to them and circumstances where they go. Um, yeah, there are similarities. We could go to the pharmacology. Ketamine isn't working on the serotonin system. It's working on the glutamate system. Glutamate being the most abundant brain neurotransmitter, the main excitatory one. Much of what the brain does when it functions is relying on glutamate and what we call glutamatergic functioning. Um, and the ketamine, to cut a complicated story short, excites the brain through this glutamatergic action, albeit in a complicated way. Um, Classic psychedelics like psilocybin, LSD, DMT, downstream of their initial effect, which is on the serotonin system, they also excite the brain. They excite the cortex. MDMA is a little different. That promotes serotonin more generally. It sort of releases serotonin, um, and that defines its effects both in the brain and psychologically. Um, Increasing serotonin functioning seems to promote positive mood, seems to promote pro-sociality, seems to promote feelings of love. And that seems to be the main thing that MDMA does. It's sort of energizing, pro-social, and promoting positive relational feelings. Yeah. Um, uh, and its promotion of neuroplasticity, there's speculation about it. Gould Dolan's done some interesting work around that, linking it to oxytocin functioning. Let's see in time whether that holds, whether that's the main thing and not serotonergic functioning, not something through serotonin. The classic psychedelics seem to promote their neuroplasticity, anatomical neuroplasticity, uh, cortical synaptogenesis, for example, the growth of branchy stuff. They seem to do that through the serotonin 2A receptor. It's pretty well established. There's some anomalies in the literature, but the weight of the evidence is saying this serotonin 2A receptor highly expressed in the cortex um, seems to be key for that. And actually, there's some interesting evidence around the 2A, serotonin 2A receptor being important generally for growth, I would say. Not growth of the body, but brain growth. Yeah, so early on in the brain, actually in utero, 2A receptor stimulation promotes the proliferation, the growth in number of cells that go on to be cortical neurons. So literally, it's controlling the growth of the cortex, albeit in the utero, in the womb. So that's cool. And classic psychedelics, yeah, seem to promote in an adult brain synaptogenesis through stimulating the 2A receptors, so that's there. There's a point of some overlap between the classic psychedelics and ketamine in that I describe both as resulting in like a net excitation in the cortex, but there's an important qualifier here because really we should understand that effect as I'd call it an excitatory dysregulation. So a bunch of jargon there. What do I mean? Scrambling brain activity. I call it the entropic brain. Brain activity becomes more unpredictable, more diverse, more varied, hard to predict across time. There's more to it. There's more to it. And just to unpack that a little bit more, to intrigue people, hopefully, the entropic brain hypothesis that I put forward first in 2014 and then updated in 2018. I'm going to update it again imminently. But it says in a very glib way, I'm going to say it glibly now anyway, more in the brain, so more bits of stuff going on, more in the brain, more in the mind. So there's a mapping between that increase in the entropy of brain activity and the richness of content of subjective experience. More in the brain, more in the mind. And you can flip that, less in the brain, less in the mind. So when something drops brain entropy, like an anesthetic, or you drop off, fall asleep, entropy drops, and of course mental content drops. Actually, if you go into dream sleep, brain entropy comes back up, and lo and behold, mental content comes back up because you're dreaming. So it's a super reliable principle. It's really simple. It sounds complicated. Just saying entropy sounds complicated. But underneath the hood, it's kind of simple guiding principle, heuristic, you might call it, like a rule of thumb. Yeah, more in brain activity, more in conscious experience. That's the hypothesis.
I love it. Thank you for sharing that with us. And we'll be on the lookout for your next update of that. I'm glad you brought up entropy because it was another thing I was going to ask you about because along with neuroplasticity and the snow globe, one of the other things that I always associated with you was work on describing psychedelics effect on the default mode network. And before I heard it in association with you, I had never, I've gone through med school residency fellowship and had never heard of the default mode network.
Mm.
And so would you mind describing to listeners what that is and how psychedelics can affect it and how maybe that if there's any interplay with what we've been discussing with neuroplasticity and entropy?
Yeah, well, I suspect there is an interplay. Yeah, and so the default mode network was coined in 2003, I believe. A default mode of brain function was coined in 2001 by American neurologist Marcus Raichle. And he was looking at patterns of brain metabolism initially through what we call positron emission tomography or PET scan studies. And he was seeing that there's a pattern of high metabolic activity in the brain. Later on, he called that the default mode of brain function. But what was key was he also noted that pattern dips when you do stuff. Yeah, when you're asked to do a task, almost any task that you're asked to do, some kind of cognitive task that taxes the mind and brain a bit, demands some cognitive resources, will dip metabolism in this same pattern. So it's a pattern that ordinarily is really high, really metabolically hungry, yet the same pattern is where you see the dips in activity when you do stuff. So he kind of put two and two together and said, well, maybe spontaneous mind activity, if you want, the default mode is highly active, uses a lot of energy, and maybe it relates to this sort of background, daydreamy style of cognition. When you do something, you're just doing a specific thing, and other parts of the brain will engage. People used to say light up, but that's kind of a bit annoying, I think. They engage, they become more active. But the default mode dips down. It's like you pulled from this reservoir and pushed it into some other aspect of the brain to do the thing. And so that was the realization first made in 2001. And then we realized, or, cognitive neuroscientists, I think it's Michael Grecius, published a paper in 2003 that had, I think, the default mode network in the title, or at least the abstract. And he was looking at how brain activity seen through fMRI, functional magnetic resonance imaging, which was really taking off at that time. It was only about 10 years old, really, in terms of being on the scene as a tool in cognitive neuroscience. He noticed how activity goes up and down synchronously across time in this pattern. And so he surmised, it's a network of functionally connected brain regions that might be spatially remote or in different parts of the brain, yet temporally synchronous and presumably functionally synchronous. Its bits of brain region go up and down together, like synchronized swimmers or something. And so then he called it, well, it's a network, and it's the default mode network. And that's what stuck around. Then we fast forward to 2012, at least when we published our work with psilocybin. It was the first fMRI work with psilocybin and the first look in the brain with fMRI at ongoing brain activity under a psychedelic. So sort of state level look at brain function under a psychedelic, published in 2012, neurocorrelates of the psychedelic state, we called it. And there we saw the default mode network bits became desynchronized under psilocybin. Instead of going up and down together like they do, in a kind of slow way, maybe, activities rising up over a few seconds and then dropping down over a few seconds and then rising up, the different bits that ordinarily go up and down like that became desynced. They were going up and down in an uncoordinated way. So that was the first insight. We also saw drops in blood flow and a hint that there was a change in activity, at least early on in that system, actually drops in a readout, you might say, of brain activity in that same pattern under psilocybin as well. And for a period, I'll just spell it out, for a period, I think we were off the scent in telling the world that psychedelics drop brain activity in the default mode network. It took a few years, but I updated how I communicate that. I wouldn't say that psychedelics drop brain activity in the default mode network. Some colleagues might, but I think it's a bit misleading. What they're really doing is dysregulating activity. In the default mode network, yes. And maybe that relates to some of the most interesting subjective effects of psychedelics, like ego dissolution. And that idea, I sort of padded out first in 2010 in a theoretical paper with Carl Friston that proposed that the default mode network might relate to our sense of ego and ego functions, or sense of self, if you want, and what the self is for and what the self does. Um, yeah, but yeah, that sort of dysregulation of activity in the default mode network plus plus, meaning not just the default mode network, remains a very reliable insight or principle about how psychedelics work in the brain, dysregulating ordinarily well organized high level systems in the brain.
And it's interesting that you brought up that it is, or at least you hypothesize that it is related to the sense of ego because, well, one, that was the second most voted thing that people wanted to hear about you from was ego dissolution. So
Yeah.
thanks for getting that in. But I had, and perhaps maybe misunderstood that the default mode network was related to like the monkey mind or like the rumination that we might have. Is that something separate?
No, I think that's valid. Yeah, there is a relationship, it seems, between default mode network activity, overactivity, and excessive rumination, for example. The default mode network seems to engage most reliably when we're engaged in introspection. That might be, ruminating in someone with depression, negative rumination. But it might also be imagining stuff. It might be like fantasy. It can have, when we daydream, the default mode network is engaged. So I think these days it seems clearer that it's not, we would say, valence specific, meaning if you're in the default mode network, you're in a negative state. That doesn't really hold. It's more that if you're in the default mode network and really engaging that system, you're likely in your head. It's just when you're in your head,
Yeah.
you could be thinking about good stuff or bad stuff, so to speak. Yeah.
Okay, that's very helpful. And to kind of round out all of these things that we discussed, neuroplasticity and entropy and the default mode network and everything, regarding how this looks for the average person who might be listening and saying, how does this have the potential to improve my mental health state? You discussed how a maybe depression in the amount of gray matter and all that may lead to disturbances in mental health. And then you also said something like ketamine that can lead to neuroplasticity, but also pairs best with psychotherapy is helpful. So if you could kind of give us a little bit in just overall what the research is showing in how this neuroplasticity can be advantageous to those who may be seeking some way to improve their mental health through one of these psychedelics.
Yeah, I think the honest answer is it's early days, but we do have a model, who've done really interesting work looking at synaptic recovery. So usually in mice, a mouse or mice undergo a period of stress, chronic stress. It can be seen that it's atrophied the brain, it's atrophied, say, cortical neurons, the branchy stuff of cortical neurons. Then, in one of his studies, he gives ketamine, and he sees that those branches that have atrophied are those that will spring back after the ketamine. So it's like a regenerative effect. So that's got a lot of people excited that that might be what psychedelics do. And I think he may be onto something. And there are some findings that we've got in human brain imaging that speak to similar principles. So, for example, we've seen in people with depression, brain networks can become quite segregated from each other. I mean, they are ordinarily, they're quite functionally separate and distinct. But that modularity might be a bit elevated in depression. But what we've seen with psilocybin therapy is that separateness between systems, that kind of segregated quality of organization of brain networks, brain systems, actually decreases after psilocybin therapy for depression. I'll put it another way, the brain looks more globally interconnected after psilocybin therapy for depression. And the magnitude of that, we call it a decrease in modularity or increase in global integrity, correlates with improvements across a couple of studies. And now, actually, in a third study of healthy volunteers that's available as a preprint, where we see that effect, we see people improve in their mental health, their well-being goes up. So, yeah, it remains somewhat speculative, but I can see a relationship there potentially between the preclinical work and this synaptic recovery, albeit mostly shown with ketamine, Connor Liston's work, and a kind of homeostatic reset or recalibration of brain dynamics after psilocybin therapy for depression. So, yeah, I am taking us to a frontier here. Might be quite hard to follow, but that's where the science is. And the fair thing to say is it is pretty young. You know, we're scratching the surface, but there could be a principle there around recovery or regeneration. You know, and it does speak to intuition, I think, about what people say subjectively. Like, what is the experience of recovering good mental health, good functionality in the world after psychedelic therapy? Well, it feels like a regeneration. It feels like a recovery of something. Um, and that isn't necessarily how it feels with other interventions for mental illness. I'm not sure that it really feels like that with, say, a treatment with an SSRI for depression. That might feel a little bit more like a management strategy, helping you get through. Um, but psychedelics seem to have the quality of being regenerative in some way. I know that's loose, that it feels regenerative, but watch this space. It may have a biology to it. Yeah.
Very cool. Well, thank you so much for sharing. First off, thank you for all your pioneering work in across the years, and thank you for sharing with us. And could you let people know where they can keep an eye on what you're doing so that they can find out the latest when your new theories come out and all that fun stuff?
Sure, yeah, thanks for asking. Well, I've got a website, carhart-harrislab.com, Carhart-Harris Labs, all one word. And on the landing page, people can subscribe to a newsletter that I put out, and there I give updates on all sorts of things, including news about a forthcoming book that I have called the working title, at least. Let's see if the editors, change it or try to change it. How Psychedelics Work, and then the subtitle, Illuminating the Hidden Mind. Yeah.
Oh,
Yeah, right. So, let's see if they try and change it. But that's
it isn't the branchy bits. That is not
right.
mentioned.
Illuminating the crunchy bits. I don't know. Yeah, but I talk about the branchy bits in there. I'm always playing with analogy because bottom line, you want the public to understand what you're going on about. So I take that really seriously, and I do my best to make it accessible and understandable to the broadest possible audience. It's quite a challenge, but the intention's there at least.
Well, I'm sure our audience very much appreciates it and just appreciates you coming on and sharing with us. And so again, very grateful for having you on and very much looking forward to the book, whatever it is called. We'll definitely be reading that.
Oh, my pleasure. Thanks for having me on, Lynn Marie. Always a pleasure.
Ah, thanks, Robin. All right, for everybody else out there, until next time.